Good afternoon. Delighted to be here. And actually, what I'm going to talk about is why genes matter in autism research. And I'm going to start, though, by giving you two stories that have nothing to do with autism. So the first, I want to talk with you about high cholesterol. So high cholesterol affects about 73.5 million people in the United States. It doubles the risk of heart disease. And about 1 in 4 deaths in the United States are a consequence of coronary artery disease. So what I want to tell you is that there's a very, very rare form of this caused by a very rare genetic mutation that leads to something called familial hypercholesterolemia. Runs in family, has recessive inheritance, meaning both the mother and the father have to contribute one copy of this rare bad gene, and you can find that in about 1 in a million families. So, in 1973, Michael Brown and Joseph Goldstein studied these very rare families, and they figured out what the receptor was for the bad form of cholesterol called LDL. So they found out how the cell recognizes that, brings it into the cell, and they worked out the pathway, how it's metabolized, and how it maintains a balance of cholesterol in the blood. A few years later, 1979, Akira Endo found, looking at some fungal extract, that there was a chemical that blocked one of the enzymes in that pathway. And he knew, because he understood the work of Brown and Goldstein, that it would make sense that if you block that it would lower cholesterol for everyone, regardless of whether or not they had the rare mutation. That was the first statin. Statins now are prescribed in more than 30 million people worldwide and dramatically decrease the risk of coronary artery disease. Second story, chronic myelocytic or myelogenic leukemia, a rare form of cancer, very deadly. The survival rate in the late 1990s, as late as the late 1990s, was about 30% 5-year survival. So a very bad cancer to have. As early as 1960, 2 researchers, you can see up there, Peter Knoll and David Hungerford, found that there was a small, you can look all the way over on the right, there was a small bit of chromosomal material in these rare families, about 5,000 families annually have this kind of cancer, and they found that there was a very small unusual chromosome there, and that led to work to try to figure out what that small chromosome was. And I've given you a bit of the history here. The key point is that what they found is that right where that chromosome came together, it was a piece of 2 different chromosomes and brought together 2 genes that were not supposed to be next to each other, creating a brand new gene that was driving forward cancer. That discovery then led to the identification of a compound that could stop that bad gene from working. It's called Gleevec, and it's transformed the treatment of this cancer. So by targeting the rare genetic mutation, they've gone from about a 30% survival rate to something now, the latest studies show about a 95% response rate to this rare form of cancer. So what I've tried to demonstrate, and I'll get to autism in a second, is that in both of these conditions, in cancer and in coronary artery disease, starting with a rare mutation has led to really 2 different kinds of paths forward to try to take advantage of those. One, in the case of coronary artery disease, was just a broad illumination of biology, and in the other case, it was a specific drug target. In the case of cancer, we knew exactly where to try to hit the abnormal gene. And that led to 2 different kinds of therapies, but both very important. One is a generalized therapy that's treating 30 million people based on a finding from a 1 in a million family. And on the other side is a precision therapy, the first really precision medicine that was marketed for cancer. So why is this important for autism? So we know that genes play a major role in the etiology of autism, the cause of autism. But we don't, at this point, have highly effective treatments, either generalized treatments or precision treatments, at least somatic treatments for kids with autism. And it was the hypothesis that led to the formation of my lab, and I think it's shared by many people in the field, that it's a lack of insight at the level that I've just showed you, at the level of the molecule and the cell and the chromosome and the brain, that has been a fundamental impairment to developing the kinds of treatments that are transforming other areas of medicine. For a long time, it was extremely difficult for us to find genes related to autism because it's not a simple genetic disorder. So in fact, we have been looking for a needle in a haystack. And actually, I have it on good authority that it's tougher than looking for a needle in a haystack, that there are less than 3 billion bits of hay in your typical haystack, and we have 3 billion bits of information in the human genome. So there are three things, three major factors that have come together to truly transform the genetic understanding of autism. One has been dramatic technological advance that allows us to read the genome quickly, cheaply, and at high resolution. The second, tremendously important, really one of the first places where advocacy, the NIH, and philanthropy came together in a highly effective way to force scientists to share data and to share biomaterials. That's been absolutely essential for all the progress in the last several years. And then the third was the discovery that Dr. Insel pointed out, that rare mutations, just like the kinds of rare mutations I talked about before, are relevant for autism. These are just a handful of papers over the last 2 years that have brought all of those together, large samples made possible by philanthropy and advocacy, using new technology to read the genome and looking for rare mutation. These are from multiple groups, from MIT and Harvard, from UCSF, thankfully, from University of Washington, from Cold Spring Harbor, all reaching the same answer and all laying out in unprecedented detail the genetics of rare mutations that contribute to autism. This slide now, that's Stephan Sanders, brilliant junior faculty now at UCSF, who was formerly in my lab. And this is a paper which will be coming out soon that categorizes the mutations based on our confidence that these are related to autism, going from greater than 99% down to 90%. But I have to tell you, having been in this field for 20 years, simply being able to show this slide is breathtaking. 15 of the 20 years that I've been in the field, I couldn't show you a slide at all. And now to be able to stand here and show you 60, 70 genes that we have confidence in, particularly on this left-hand side, is transforming the field. So I'm going to show you a couple ways that it's transforming the field. One is I'm going to take you back to the idea of finding a personalized treatment based on a rare mutation and an abnormal enzyme. This is a paper that was done in collaboration between my lab and Joe Gleason, who's now at Rockefeller, was previously at UCSD. And we found in a very rare families, actually several families, in the Middle East, that they had recessive mutations, 2 bad copies of a gene that's involved in amino acid synthesis in just the building blocks of proteins. And what we were able to determine is that that was the cause in those families of autism and epilepsy. We were able to show that in mice, you could give them the same mutation, and if you fed them lots of branched chain amino acids, the abnormality went away. If you starved them of amino acids, which is on the left here, you would get neurological symptoms. And so it's allowed us now with IRB from Yale and UCSF and Rockefeller to treat a handful of families in the Middle East who have these mutations just with dietary supplementation in order to see whether or not we can have the same result if we start very early, because we know the children who are at risk, they're born with the mutation. The other example, Dr. Insel very graciously already introduced this idea. This is along the lines of the idea that we may be able to understand biology more broadly as opposed to finding targeted treatments because of a specific mutation. And as Dr. Insel pointed out, this was made possible, so that's Nenad Sestin on the left. Nenad, thanks to funding by the NIH, began to create a map of the molecular landscape of human brain development. So he was able to give us a picture of how genes work and how they build a human brain. And Jeremy Wilsey, at the time a brilliant brilliant graduate student, now brilliant junior faculty member, they're all brilliant. It's like Lake Wobegon in my lab. They're all like well above average. So what we did, you can't really see at the top, but what we did was we took the genes, the first genes that we found that we had very high confidence were related to autism, and we used the data that was developed by Dr. Sestin along with others in the BrainSpan project to say, what do they do? How do those specific genes, how are they involved in building a human brain? And then we could ask a question, was there any point in brain development where those genes were working together in a way that would not be expected and where there was really a convergence in their potential risk for autism? And what we found out was that, in fact, they did form a network that was much more closely tied together than we expected by chance, that took us to one particular epoch in brain development, in mid-fetal development, about week to 10, to week 24 in mid-fetal development. But it allowed us to go further than that and to look in the cortex, as Dr. Insel pointed out, exactly where in the cortex this network of genes was acting, and it was only in the deepest layers of the cortex, layers 5 and 6, and we were able to go beyond that and ask what are the specific cells in those layers that have this network of genes, and we were able to find that they are what are called excitatory projection neurons, a particular subtype of neuron that create the long-range projections from the cortex to other parts of the brain. So, what we have, I think, is just the beginning of the kinds of stories that I told you about coronary artery disease, hypercholesterolemia, and cancer. We are just in the earliest stages of that progress, but as Dr. Insel pointed out, it is breathtakingly exciting. We do have a roadmap. We do have a fast car. There are some turns along the road as well, and I'll just point out a couple before I end. So this is a picture taking the genes that I showed you that we all now in the field agree have very high degree of likelihood that they're involved in autism or are definitively established in autism, and to try to see sort of how they fit together and what kind of processes they involve. And the only thing that you need to get from this diagram is that it's really complicated. There are lots of genes, and they're doing very important basic things in the cell, and we have a long way to go in order to work out exactly how that plays out in brain development, but we're beginning to get the kind of traction that I showed you on the prior slides. And then the last point that I'll make out is that, we started with the idea that we were looking for genes for autism, and we found genes that dramatically increase the risk for autism, 30-fold increases in risk for autism. And then we go back and look and find out that the very same genes are being found by our colleagues who are studying intellectual disability and schizophrenia and epilepsy and congenital heart disease and metabolic disorders. It's not complete overlap, but what it begins to suggest is, as Dr. Insel pointed out, these are developmental disorders, and the abnormalities are affecting fundamental processes in development, including in brain development. So, while we have work to do, I'm going to end just by saying that it is really, truly a new age in gene discovery. It is not hyperbole to say that 3 years ago I could not have stood here and given you a list of genes that we had, that the entire field shared confidence in were related to autism. We have potential avenues to develop new treatments, both through illuminating basic biology and by providing opportunities for precision medicine, which always come as a surprise, but they're there. The challenges that we face now are to continue to find these clues and to understand what they mean. And my take-home point is that while people think about genetics, and they often think about how much does it explain, or is it a diagnostic tool, and in fact, I would make the argument that what genes can tell us about biology is far more important than whether or not they're going to tell us that someone's at risk, because it's going to be through the understanding of that biology that will transform the field. The happy notes are that the examples I gave you at the beginning were 30 years from discovery to the development of a treatment, but the field is moving at light speed for reasons that I've described here. Our ability to study brain circuits, to understand brain development, to manipulate and edit the genome, and to do large-scale genomic studies are unprecedented, and that there's no question that's going to shorten the time between our discoveries and the development of novel therapies. What you can do, what you need to do, anyone who has the capacity to do so, is to enroll in clinical trials. We can do nothing, absolutely nothing, without patients and their families contributing to our work. You know, you can see the commercial message. I think it's very important to support basic science, that it's through basic science that these novel insights and potentially novel therapies will arise. Thanks very much.